Hello, my talk is titled, Can We Get Something for Nothing? Physiological Information in Resting State fMRI. I'd like to thank the organizers for inviting me. As you've heard in the earlier talks, there's such an abundance of resting state fMRI data and many ways to improve our ability to detect neuronal information from it. You've also heard of ways to better understand and characterize the cerebrovascular contribution. So building on this knowledge, what can we extract from the resting state fMRI signal in addition to brain connectivity? Is there information that's free for the taking in this noise contribution? So just to recap of the physiological noise sources in resting state fMRI, these mostly come from respiration and cardiac pulsations. They could lead to changes in intravascular CO2, head position, and in magnetic field inhomogeneities. To spell them out more explicitly, each source of the noise also exhibits different frequency content. They're very different from each other and from that of thermal noise. Respiratory noise exhibits time-locked frequency of around 0.3 hertz and a low frequency variation of about 0.03 hertz. The influence on the both signal are through field changes, CO2 changes, and oxygenation changes through blood flow. Cardiac noise shares some of these mechanisms, but its frequency signature is different at about 1 hertz for the time lock portion and 0.01 hertz for the low frequency portion. So both sources of bodily processes can also induce head motion, which also has a non-physiological contribution, of course. So why is this important? Well, the fact is that in resting state fMRI, the noise is larger than the signal. To visualize this spatially, regions of high vascular content also tend to exhibit more of these noises, notably in the primary visual and sensory areas. Of all of the sources discussed, respiratory noise, including CO2 and respiratory volume fluctuations, accounts for the largest source of physiological variance in the low frequency fMRI signal. To further clarify respiratory fluctuations, we can measure end-tidal CO2 or PET CO2 as a surrogate for intravascular CO2 fluctuations, which is arguably the largest source of physiological contribution in resting state fMRI. This was first demonstrated by Richard Weiss and colleagues and later refined in Katie Chang's work, showing the strong influence of CO2 variations on the fMRI signal in cortical gray matter. If your goal is to remove the CO2 contribution, then mechanical clamping of end-tidal gases might be the most effective way, using a Respirac system, for example. As you can see here from our own data, this yellow CO2 spectral peak is completely removed under CO2 clamping. So we then investigated the difference between clamping and free breathing in terms of resting state connectivity. So under clamping, as opposed to free breathing, the resting state fluctuation amplitude, RSFA, and the global brain connectivity, WGBC, both decreased. And to illustrate this graphically, so this is connectivity for 3 major brain networks: the default mode, the motor, and the visual network. And as indicated by the orange color, connectivity is a lot higher in free breathing compared to clamping. And more importantly, clamping is the most effective way of removing the CO2 response or effect. It's more effective than any post-hoc CO2 correction strategy. But if you don't have a Respirac or similar, is there a way to achieve the same effect for free? So for that purpose, we investigated the global signal regression approach, white matter CSF regression, and a combination of the two. And it turns out that white matter CSF regression results in something most similar to that of CO2 clamping, as you see in the middle row. And so what this says is that you can potentially achieve pseudo CO2 clamping for free. But to go back to this physiological response in resting state fMRI, our group further mapped the amplitude and the latency of these response functions. So not only for CO2, but also for respiratory volume and for cardiac variability. So both CO2 and respiratory volume fluctuations are most strongly reflected in the cortical fMRI signal, notably in the primary visual and sensory areas, but also in parts of the default mode network, namely the precuneus and the prefrontal cortex. For the heart rate variability response, the hotspots are near the ventricles in deep white matter. So given this robust CO2 response, we wondered if we could actually get CVR for free from resting state fMRI. And in our own data, heart rate variability explained on average about 8% of the fMRI signal in resting state, and respiratory variability about 6% when it's orthogonalized from the CO2 effect. So the CO2, which is remaining, accounts for as much as 15% of the resting state fMRI signal. So there is promise. And so it's tempting to think that PET CO2 can offer us an inroad. We can use it to assess quantitative CVR if we measure it. So Kevin just spoke about different CVR mapping strategies, and different methods imply different levels of patient effort and safety, ranging from CO2 targeting, manual gas delivery, breath holding, deep breathing, to all the way to resting state on the far right. So because of the different levels of CO2 changes involved, the actual quantitative values of CVR and their spatial distributions from these various methods may not be equivalent, and it's very important to recognize this point before going any further. So the idea of extracting CVR from resting state fMRI is not new. It was first seen in 2014 in Sridhar Kanyapati's work, in which resting state fluctuation amplitude was proposed as a qualitative surrogate for CVR and compared against breath hold CVR measurements. And there was a significant correlation, but only in the younger subjects. Another qualitative CVR metric is the coefficient variation approach proposed by Jahanian and colleagues. This was again validated against the breath hold method successfully. One question here about both of these methods is whether it's safe to assume that the resting state signal is dominated by vascular effects, which may be questionable. So later, Liu and colleagues proposed a third qualitative CVR approach based on regression against a portion of the global signal, specifically the portion that is varying at the frequency of end-tidal CO2. So unlike the voxelized fMRI signal, the global signal may be more likely dominated by bulk physiological effects. So the global signal approach was successfully validated against the hypercapnia approach for within subject spatial variations in qualitative CVR. So later, as our group has been in the habit of recording end-tidal CO2, we investigated the feasibility of quantitative CVR measurements using resting state fMRI. So this is just an extension of the earlier work on estimating the response functions. So the area under the response function is actually by definition the quantitative static CVR. And so our results demonstrate not only qualitative but quantitative agreement between the resting state quantitative CVR method and the hypercapnia method. So these are two sample data sets from healthy adults. But so far, we've been mostly talking about just a single CVR values, otherwise known as static CVR. But more recently, Procopio and colleagues were able to obtain a more robust estimate of the CO2 response function from resting state fMRI, also referred to as dynamic CVR. So as demonstrated here, there is this difference that was shown between the dynamic CVR induced by task-based fMRI compared to resting state fMRI. So this method definitely makes the estimation of quantitative CVR in resting state more feasible. But what if you don't have end-tidal CO2 recordings, which is required for quantitative CVR estimation? What if you only have bellows measurements? So the relationship between these two is not linear and might vary with time. So some recent work from our lab demonstrates that, yes, it is actually possible to generate CVR tracings from respiratory belt tracings using machine learning. So this work is under review, but it shows a lot of promise. So again, we're kind of scaling back the things that are required to get more out of your resting state fMRI signal. So to summarize, it is possible to achieve CO2 clamping effects for free by using white matter CSF regression. It is also possible to obtain quantitative CVR for free using end-tidal CO2 recordings. But worst comes to worst, if you don't have end-tidal CO2 recordings, you can actually get it for free, potentially by from respiratory belt recordings. So there's still a lot of work to be done, but there's definitely promise. Now to turn our attention to the CSF signal, so cerebrospinal fluid, another physiological noise contribution. It exhibits 3 clear frequency bands, namely at 0.9 to 1.2 hertz, which is in agreement with heartbeats, 0.2 to 0.5 hertz for respiration, and a low frequency band, which is 0.01 to 0.03 hertz. So CSF convection in the low frequency has been reported as a force driving lymphatic flow, and there are several mechanisms of CSF change in fMRI. This has been investigated by several groups over the years. One is through partial volume changes with tissue because of the pulsatile nature, either through cardiac or respiratory fluctuations or through brain activation. The other main mechanism is through inflow. So CSF inflow has been known to change during the sleep cycle. So to discuss it in more detail, the signal origin differs in different CSF compartments due to their different flow patterns, as you can see here. So more likely through partial volume effects is the lateral ventricle, because there the flow is more in plane. Whereas for the third ventricle, fourth ventricle, and the aqueduct, the signal origins are more likely to be inflow. So Retama and colleagues showed the clear influence of cardiac and respiratory fluctuations driving the CSF fMRI signal, as you can see here. So on the left-hand side, they illustrate the signal fluctuation amplitude in these 2 different frequency bands for respiration and cardiac. And correspondingly on the right side, they show the percent variance explained by these 2 respective physiological signals in the brain. And definitely for cardiac, there's a lot of variance explained by the CSF signal, in the CSF signal. In our recent work, we further dissected the CSF by compartments. And so you can see here, these are the power spectral densities for different tissue and CSF compartments, namely the lateral ventricle, third ventricle, gray matter, white matter, and arteries, veins, and aqueduct. And the thing to note is that so the high frequency cardiac peak is evident in the third ventricle and the aqueduct, but not in the lateral ventricle. So the low frequency CSF signal is also of great interest. It is found to reflect low frequency blood pressure variations usually observed using pulse oximetry. So you can see it here. So there is this low frequency signature. And on the right-hand side, we plot a series of pulse finger plethysmograph derived measures of dynamic cardiac or vascular fluctuations. And this last one, called the low frequency pulse intensity ratio, has been established as a surrogate for dynamic blood pressure. And so this is reflected in all of the CSF compartments, but definitely also reflected to some degree in the tissue and vascular compartments. So this is the distinction between the CSF and non-CSF signals. And lastly, recent work from Yunjie Tong's group and from Laura Lewis's group show the strong influence of sleep on CSF flow. And so this is an example from Yunjie's paper showing that the two can be linked by a time derivative relationship. So the CSF pulsation frequency in deep sleep is lower than the awake state, and the cause of the CBV fluctuations in the awake and sleep states may actually be different. So this is actually very intriguing, using the CSF flow pattern as a way to detect awakeness. So again, to summarize, so the research on CSF signal in fMRI is just starting, and we already get something for free. So the CSF signal due to inflow can be used as a surrogate for cardiac pulsation, but not all CSF compartments are created equal. In other words, not all CSF compartments exhibit this. For example, if the flow is more in plane, not so much up and down, then we may not see this through plane inflow effect as much, as is in the case of the lateral ventricles. And the low frequency CSF signal can be a marker of dynamic blood pressure, so lymphatic flow, and even CBV fluctuations, so blood volume fluctuations. And this can even be used as an indicator for awakeness. So there is a lot of work to be done, definitely. So the theme of this talk is to get something that we usually would discard as noise, and the moral is definitely, yes, we can. We have to do some very careful signal processing, but there are lots of interesting markers that are kind of hidden in this resting state signal that we're still unearthing. So I would just like to acknowledge Ali and Ali Reza, who contributed to some of the data that I showed in this educational symposium. And I would like to thank Stefano, Molly, and Cesar for organizing the symposium, and finally acknowledge my funding sources from Canada. So thank you very much for your attention.